Thank you. Can you guys hear me? There's a lot of... All right, I'm loud. That's fine. So we're going to talk about seagrasses in transition, and we're going to shift focus from the tropics to the temperate zone a little bit. And we are a transition zone because although we do have Zostera marina, or eelgrass, which is the dominant species in the northern hemisphere, we also have Halodule wrightii. And what makes this interesting, and there's also some Ruppia, but Ruppia gets no love. So what's interesting is that Zostera and Halodule have very different distributions, right? So we have Zostera, which is the dominant species in the temperate northern hemisphere, and then you have Halodule in the orange here, which tends to be found more in subtropical to tropical regions. And the only place that you find the two of them co-occurring together in the western Atlantic is right here. And so this is the U.S., this is southeast, and this is North Carolina. So D.C., there, and then we have North Carolina. And that's the only place where we have in our part of the Atlantic where the two co-occur. And what's actually interesting is that this means that Zostera is at its southern limit. So we are sort of at the front lines of climate change when it comes to our temperate meadows in the U.S. And so because of that, we are seeing some negative impacts of temperatures in particular. We can actually see some areas, for example, here's Phillips Island. It's a study site that's been followed since the 1970s by NOAA, the National Oceanic and Atmospheric Administration. They've followed this site pretty regularly with some of their reports. Here's what it looked like in 2008. Not super pretty, but very healthy, very stable. And we go back 8 years later, there's very little grass. If you go back this year, there's really nothing there. But besides that, we also have some meadows that are actually stable. So we do have some meadows that are actually maintaining their area at least. And then we actually have some areas that are actually increasing. And that's particularly due to Halodule and Ruppia expanding, but we are seeing all three sort of states. So we are seeing this transition zone. We are seeing multiple responses to our current climate conditions. And so what we're trying to figure out is that why are some of these meadows more resilient than others? Because it's not just a species thing. We're not just seeing, oh, the meadows that have Halodule are doing better than the Zostera. You're kind of seeing, there's not a definitive pattern for why some are more resilient than others. And since this is a conference about putting science into action, we were also contacted by our local managers trying to figure out how they can best manage this resource. And they don't particularly care about seagrass for seagrass sake, but it is a really good fisheries habitat. And so they want to know, why are some of our meadows declining, especially those that are important for commercial or recreational fishery species? So to help answer that question, we established a monitoring program. But this monitoring program was set up so we could look at not just the structure of what's there, but also potentially the resilience. So here we are in the U.S. So here's North Carolina, southeastern part. There's Florida. We're in southeastern North Carolina, and then this is the southern limit. I know people from Virginia, Bob Orth, like to say that it's very close in Virginia, but we actually have the southern limit in the western Atlantic in North Carolina. To get sort of studying those interactions between climate and our seagrasses, we picked 3 sites. Back Sounds, if anybody is familiar with Judd Kenworthy or Mark Fonseca's work, that's where a lot of their stuff happens. The New River, and then also Topsail. So these 3 areas were selected because you have all 3 species, and you have a wide variety of estuarine habitats. So here's an example. Within our 3 regions, we set up 3 monitoring sites. The green on this map are the seagrass meadows, which are mapped by the state about every 5 years. Within each of our 3 major regions, 3 sites. Each site has 3 transects. And we ran the transects perpendicular to shore because this is the protocol that the National Estuarine Research Reserve System uses. They have 29 reserves located across the United States. So we could directly compare our data to those other reserves to see how they're responding. These transect lengths vary, transects vary in length between 50 to 300 meters, just depends on how deep the meadow goes out. Each of the red stars there show you where we also have a paired water temperature and then also a light logger, so we get data every 15 minutes. So what do we do with these transects? So we go April through September, which sort of incorporates the time when Zostera is at its peak, and then also when Halodule or Halodule are at their peak in September. We do typical structural metrics. So we lay out our transect line. Every 10 meters, we get % cover by species and total seagrass. We also collect biomass cores, and then we also collect area data using drones. And so Kenji is actually going to talk about some of the successes and some of the problems we've had with using drones to map our areas later this afternoon in session 5A. So in addition to the structural metrics, which are pretty standard across a lot of your seagrass monitoring protocols, even globally, we wanted to add some physiological metrics and some metrics of resilience. So in addition to the quadrats. We also collect plants and look at carbon and nitrogen ratios. We measure productivity twice a year, and then we also collect cores, look at seed density and viability. So we're looking at viability so we get an idea of what's the actual functional seed bank. So not just the number of seeds that are there, but the seeds that could actually germinate. And so these are the metrics that we kind of want to, we're throwing sort of everything at the managers right now. We're collecting this data over 3 years, and the idea is that we can then narrow that down to a few parameters that the state can actually use to monitor these sites. They don't have enough manpower to do all of this monthly from April to September, but they could do a couple things. And so we're trying to figure out what gives us the best response. So right now I'm going to give you some of our preliminary data. We're going to start with our water quality data. So this is the daily mean water temperature from all 3 sites from July of last year through December. What's interesting is that all 3 sites have very similar water temperatures, despite the fact that they have very different connections with the ocean, and they have different hydrodynamic regimes. What's also interesting is that, again, we have Zostera, but I'm going to put this line here at 28 degrees C. These are daily mean water temperatures, right? So our Zostera is experiencing daily mean water temperatures above 28 degrees C from about July, when we started monitoring, all the way through September. And there are definitely some times where it's down a little bit lower. But we picked this metric to show you that this is a temperature that when they get sort of above this for any period of time, Zostera tends to get very stressed. And you also start to see large-scale declines. In fact, we've seen and documented several large-scale declines just north of us in Virginia when water temperatures were above 28 degrees C for just 2 weeks. And we're seeing this for multiple months. And if you go into September, daily mean water temperature was above 30 degrees C for about 2 weeks. So we wanted to see, well, how does Zostera respond to that? Well, we can give you some information about what we expected Zostera to actually respond and how that would interact with Halodule, because these are the sites that Judd Kenworthy used for his dissertation research in the 1980s. So we can actually go back and directly compare our results to some of the NOAA data from that time period. So this is Judd's data. He has down here, time is on the x-axis. Now this is leaf canopy surface area, which is not a direct proxy for percent cover, but the sort of magnitude of the trends will be similar. So here we have the white bars are Zostera, the solid bars are Halodule. You can see in North Carolina, between March, April, June until July, Zostera has the dominant surface area, and that's when water temperatures are cooler. Okay, so as they start to warm up in the summer, which we have shown in our data, you sort of switch. So Zostera is now no longer the dominant species in the area. Now you have Halodule is the dominant, and that maintains through the later summer months into the autumn, and then only once you get to January does Zostera kind of kick back up. But I want to point out that even in October, they found Zostera. So what did we find after seeing those daily mean water temperatures that were above 28 degrees C? So we're going to talk about our transect data. We're averaging that data across one of our sites, all the entire transect. So we're averaging our data across 200 meters. And what do we see? So now we have blue is Zostera, the orange is Ruppia halodule. So we do separate out Ruppia in our biomass. It's really hard to do that in our quadrats. It looks very similar to halodule. So we actually have to only separate that out in the biomass cores, but for the transects, they're a combined complex. So here we have percent cover, and we're looking at July, August, and September. And so originally we kind of thought, well, with the warmer water temperatures, Zostera is going to decline earlier in the year. Okay, so you still see it, but we'd expect it to decline earlier than what they saw in the 1980s, because it's a little bit warmer now than it was previously. And that's not what we saw. Percent cover was still pretty high. The majority of the seagrass within our quadrats, the cover was Zostera up through August. What's different, though, is what happened in September. We lost all of our Zostera. And this happened across every site where Zostera was found within that region. So we see the Zostera is gone. So it's not the timing of this change is happening, it's the magnitude. Well, the next question is, all right, well, we still have Ruppia halodule, we still have seagrass. What's happening? Can that replace what's going on with the Zostera? So to answer that question, we want to take a step back and actually look throughout all of the transect data. I know this is a lot to digest, so let me walk you through it. The x-axis here is the distance along the transect, so every 10 meters up to about 200, still percent cover. Blue is still Zostera, orange is still Ruppia halodule. And you can see in the shallow part of the meadow, and when I say shallow, the max depth here is 1.5 meters. Right, so in the 50 centimeters or less part of the meadow, you see there's a lot of interaction. You have both Ruppia and Zostera. They're found together in that area. Once you get a little bit deeper, where there's a little more light limitation, you get to about a meter's depth. From 90 meters on out, you really don't see any Ruppia halodule. At that point, you just see Zostera. So there's a lot of blue at the end. Now there's multiple bars here at each distance, and those are just the different months. And so what I want you to see is that in the shallow part where you have Halodule Ruppia, we have 3 bars. We had grass there every time. But once you get out past 100 meters, there's only 2 bars or less, which means that in September, half of this meadow was now gone. We lost everything from 100 meters out. So even though it's still a meadow, it's still functioning, it is now decreased in area quite dramatically. And we saw this across all of our sites. So there is some transitioning happen here. We do see Zostera. What we're not sure is when it disappears or when it sort of declines due to warmer water temperatures, how does that affect the function of the meadow? How does that affect the resilience of the meadow? And that's what we're hoping to get out with the remaining part of our the next 2 years of this project. I do want to point out that we came out in April of this year and Zostera was back out to 200 meters and it was 60 centimeters long. So it is coming back. It's just during that time period where it's absent, we're not sure what happens to those meadows. So in conclusion, we see that our Zostera is regularly exposed to temperatures greater than 28 degrees C, and those are daily mean water temperatures. And we do see that in September, at least this last year, there was a 2-week time period where they were exposed to daily mean water temperatures greater than 30. As a response, we see that Zostera did decline, but the meadow is maintained. So it's different than some of our more temperate meadows where they only have Zostera and Ruppia, whereas once the Zostera is gone, the Ruppia is really exposed to a lot of physical energy, and that tends to disappear as well. And so because we have the Halodule in combination with the Zostera, means that we actually maintain the meadow, but how does that affect the overall size is also reduced. The timing of the Zostera seasonal declines was similar to the 1980s, which was a little bit surprising. Like I mentioned, we thought it would happen earlier. Again, this is just 1 year's worth of data. We're going to get the next couple of years and hopefully bring back a fuller story. Again, this is also just a percent cover. I want to get to the density, the biomass, all the rest of our actual resilience metrics. We see the extent of the loss, so the magnitude is different. So the timing is similar, the magnitude is different. You completely lose the Zostera there. And so we don't know how, we do know that Halodule does not replace Zostera in all those areas, but it does maintain that meadow. So maybe there's some facilitation happening there that allows the Zostera to come back once water temperatures do cool down. And we don't know about this sort of shifting species composition. How does that affect the meadow function and resilience? That's what we're hoping to get at with this project in the future. But that's where we are with our preliminary stuff. So with that, I'd like to say thank you to all of my student volunteers and our collaborators and our funding sources. So thank you.
So do we have any questions for Jessie?
Did you have effects of any of the hurricanes in September that could have led to part of this diminution?
We didn't have any. Matthew was 2016, so we didn't have any last year
Irma?
during that time period.
No.
Not in that area.
So it was not related to an intense storm of any kind?
No, not that we know of. Yeah, so thanks.
Hello, thank you for the talk. I was wondering if you know why you find Zostera not in deeper areas and not in shallower ones.
The Zostera marina?
Yeah.
Yeah, we do find it in shallower. It just, when we talk shallow at low tide, it's almost intertidal. So they're getting exposed. At that point, we have our loggers. Temperatures get to 36 degrees at that point in the really shallow water. So if you have just a little bit of water that's maintained, so maybe like 10, 20 meters from the shallow end, it can grow there, but then it gets too hot.
So you think the temperature is the main factor driving this?
I think so, but we have also other water quality data that we're going to go and look at and run some, because we have a YSI getting pH, salinity, DO, all the water quality parameters. We don't see a lot of grazing activity, so that's nothing else we might look into, but maybe more grazers coming in with the warmer temperatures. But to see the big decline happen after that really intense temperature that we have seen result in large-scale Zostera declines in our near neighbors, that's our leading hypothesis right now.
And just the final thing, when you go to northern latitude, is Zostera marina able to colonize these shallow parts?
Mm-hmm. Yep, but it's much cooler. Okay. Yeah, but yes, it can shallow as well.
Thank you, Jessie. I was just wondering, is your Zostera annual or perennial? So do the plants really die off, or do they persist on their below-ground biomass when the cover drops?
Right, so we did some work, part of my PhD, showing that there's a variety of life history strategies in this region. This meadow, according to NOAA, is perennial, but that's not what we saw. And when we came back this year to do our cores in April, we saw a lot of seedlings. So we do think that it's sort of shifting towards that more of that annual, relying on seeds to come back every year. And I think, we saw that in some other meadows in the early 2000s, and I don't know if it's now becoming more widespread in this area, and maybe that's what Zostera is, how it's responding. But that's another can of worms.
All right, I think we'll need to break there. Thank you, Jessie.